Okay, we are ready to continue. Just a little changing. Please, for Lisa Maria, for the speech, we anticipate her speech. So, Maria Follesa, spatial variability of conjunctives in the North Mediterranean. North Mediterranean.
Okay. Good morning to everybody. I am Cristina Follesa from the Università of Cagliari, and I'm going to show you some data relative to the spatial variability of demersal chondroites in the North Mediterranean. So, as it has been already underlined during these 3 days of meeting, elasmobranch count a conspicuous number of predators, playing an important role within the marine ecosystem. Anyway, it's well known that some anthropogenic activity like overfishing and habitat degradation have altered the marine population, especially shark and rice. At this moment, fishing pressure, as we know, on chondroites species is increasing. Recent global data show an increase of 42% of the trade in shark meat. And it's also reported that the true total catch is considered sometimes 3 or 4 times greater than the reported. This because, as we saw this morning, chondroitic catches are unregulated, often are misidentified. Sometimes the specimens were discarded at sea, resulting in a lack of species-specific landing information. At the Mediterranean level, following what's reported by the UCN, 50% of rice and 54% of shark are facing an elevated risk of extinction. For this reason that starting to 2010, the FAO GFCM has adopted measures to reduce the bycatch of pelagic species, has banned finning practices, has prohibited the capture and sale of shark and race listed in the Annex 2 of the Barcelona Convention. In spite of this, no effective management measure against the overexploitation of the elasmobranch species have been implemented, resulting also in Mediterranean in the same situation we have already seen at global level. For this reason that thanks to the availability of MEDIS survey data, in this talk we will give a standardized picture of the occurrence and abundance of cartilaginous fish coming from different parts of the Mediterranean basin, and we'll make also a comparison between these geographical sub-area. The geographical sub-areas involved in the MEDIS survey, you can see in this table, there are geographical sub-area coming from Spain, Italy, France, and Greece. Also the other country from the Adriatic level. So going to the results, we can see that during the 4 MEDIS troll survey carried out from 2012 to 2015, we collected specimens belonging to 8 orders, 15 families, and 41 species. As you can see in this slide, among the 22 battoid species, we found that Torpedo marmorata was present in all geographical sub-area investigated, while Raia clavata and Raia miraletus present a percentage of near to the 90%. Among the 18 shark species found, we recorded that Galeos melastomus and Chiliorinus caniculus are present in all geographical sub-area, while, for example, Etemoterus spinax present a percentage near to 90% another time. During this survey, we collected also some rare species, spot data, relative, for example, here you can see the list of rare species for Bathoides and also for Squaliformes. You can see that Etaomileus of Venus, for example, is captured only in the Strait of Sicily and in Ionian Seas, while for Squaliformes, Galeus atlanticus was caught only in Alboran Seas. So, except for some cases, we found in our work that Bathoides had a preferential distribution on the continental shelf, while Squaliformes were more frequent on the slope. So, going to the result relative to the occurrence registered in the whole bathymetric strata, so from 10 to 800 meters, we can find in these graphs where in the white bar represent the GFCM, the geographical subarea present in the western Mediterranean seas, the gray one those of the central Mediterranean, and the black one those relative to the eastern Mediterranean. In this graph you can see that Chilioninus canicula is well distributed along the basin, with the highest value of occurrence reported with green spotted in the map, are recorded in Balearic Island and Corsican Sea, while the lowest value indicated with red spotted in the map were registered in the South Tyrrhenian Seas and also in Ionian Seas. Relative to Raya clavata, it's well represented in all Mediterranean basin, except for the Ionian Sea, where it didn't found, and also in Alboran Sea, where we registered the lowest value of occurrence. Regarding the occurrence registered in the shelf, we found that Raya miraletus species linked to soft bottom were mainly present in the central and eastern Mediterranean, reaching the highest value of occurrence in another time in Corsican waters. Corsican waters can be considered an hotspot, a real hotspot of elasmobranch diversity. The torpedo marmorata was also found in all Mediterranean basin. Going to the occurrence registered on the slope, we found that Galeus melastomus present an occurrence of near to 90% in all geographical subarea, except for the Gulf of Lion and also another time in Corsican waters, where the value is reported higher than 90%. Temoterus spinax was found mainly in the western central part of Mediterranean, while in the eastern part this species was not recorded in Cretan Sea. Other species that present a high level of occurrence in the slope, Squalus blainville, that was mainly found in the eastern part of the basin, but in any case, the highest value of occurrence was found another time in Corsican waters. Dasiatis oxyrincus show also its peak of occurrence in Corsican waters. We try to identify also the abundance expressed as a biomass and the density in the overall strata, so from 10 to 800 meter. And for this strata, we don't found any specific trend. The biomass and the density of shark and bathoides were variable between the different GSA. Among shark, we found that Galeus melastomus and Chilioninus canicula present another time the highest value of abundance in Corsican seas, even if the value of Galeus melastomus appear double respect to of those of Chilioninus canicula. Temoterus spinax was abundant in the Cyprus waters. Among the bathoides, we reported only the main species caught. We can found Raya clavata that another time registered high abundance in Corsican waters. Raya miraletus is more abundant in Strait of Sicily, while Torpedo marmoratus was found in all Mediterranean, but with very low value of abundance. Regarding the abundance on the shelf, as I said before, apart from Scilurhinus canicola that was abundant also on the shelf, generally in this macrostrata we found a preference of the bathoids. Here are reported the main species recorded with in green the max value with the geographical subarea where it was caught, and in red the minimum value. I don't comment this data because they are time consuming. And also for abundance in the slope, as I said before, Squaliformes present the highest value in respect to bathoids. Here the list and the map with the highest and the lowest value of abundance. But in any case, in this strato we found also some bathoids, like for example Radi family with a preferential of Raia clavata and Dipterus syrincus, while the Asiatis and Torpedine family were very scanty. We performed also a gamma model for density and the biomass of both groups, so shark and bathoids, using considering the geographical coordinates, depth, and some environmental factor like temperature at bottom and at surface. In the table is reported the best model. From this model we can find that the temperature had a significant influence in the species distribution of both of elasmobranch, but with an opposite trend from between bathoids and shark. The highest biomass of shark were in fact found in the geographical subarea where we have a low value of temperature, and the opposite for bathoids. Also depth was considered one of the most driver influencing the distribution of shark and bathoids, but also in this case with an opposite trend. So in fact, from the map, the map constructed by the model, where in the upper part are reported the density, and in the lower part the biomass, we can find that the density and biomass of shark increased with the depth, and we found the highest value near to the Balearic Island, the eastern coast of Corsica, the northeast part of Sardinia, in the southern part of Sicily, and also in Aegean Seas. Regarding bathoids, the model show quite the similar geographical distribution pattern of shark, but with hotspot at lower depth. So, in conclusion, we can say that with this presentation we provide standardized and updated information regarding distribution, occurrence, and abundance of chondroites in the northern part of Mediterranean. Comparing our result with the literature present, an abrupt decrease in a number of chondroid species was found in the Northern Adriatic Sea. But in any case, in the rest of the Mediterranean, we found the most widespread species in the basin were almost the same found in a previous study conducted some years ago with MEDICE data. So we can conclude that this overlapping should be probably attributed to the higher fishing pressure resilience of the species. Even if with a comparison from F4 map, this map on the right is performed inside the European project StockMed, but with a comparison from the F4 map with our map relative to density and also biomass, we can find that some area under high pressure, fishing pressure, as Adriatic Sea, for example, register low abundance of chondroites, while other area, like for example Sicily, south of Sicily, where we have an higher fishy pressure, we found an abundance of the species. So we can conclude that the fishing pressure should be considered as one of the main variables that lead the abundance of chondroites, but not the unique. Sometimes also other environmental drivers work together to shape their distribution. I would also add that to have a very complete picture of the distribution of demersal asmobranch in the Mediterranean Sea, we have to take also in consideration some countries present in the southern part of Mediterranean, like Libya, like Morocco, Tunisia, where we have an high level of species. This work will be published inside the Mediterranean Demersal Resource and Ecosystem, a monograph that will be printed in the next December 2009 for the 25 year of Medistrol survey. I would also say thank you to all co-authors, some are present here, for the effort made in the construction of this paper. Thank you.
Thank you very much, Cristina, for this interesting speech. We have no time for questions, sorry. Thank you very much, Cristina.
So, it's a pleasure to introduce to you our invited speaker, Diana Pasmino, and just to give you a short CV of Diana. Diana got her PhD very recently in 2017 at James Cook University in Australia in the Centre for Sustainable Tropical Fisheries and Aquaculture. And from last year, so from 2018, she's faculty member of the University San Francisco de Quito in Ecuador, but also researcher at the Galapagos Science Center. And I think being in the Galapagos Science Center, of course, one of the topic or her expertise are evolutionary biology, but also conservation, ecology, and she's using genetic and genomic approaches to inform conservation. So thank you very much. And sorry, I need to give you also the title of the talk, Use of Genomics for Applied Elasmobranch Conservation.
Thank you very much. Thank you for that introduction. I will just give an overview on genomics and the use of genomics in branch research and conservation. And I know genetics and genomics can be a hard topic. Don't worry, I'm not going to focus on the technical details behind the molecular techniques, but rather I'm going to focus on the applied like the applications for conservation itself. So first, I'm going to start talking a little bit about the genetics and their importance and challenges in elasmobranch conservation, move to the shifts to genomics and the relevance as well, and finally touch on some specific cases of genomic studies in elasmobranch that have helped us respond to some questions at different levels, from species to populations and individuals. So I want to start highlighting that genetics have been very important in elasmobranch research and conservation in the past decades. They have allowed us to identify population structure, to identify management and conservation units, stocks, to assess the connectivity between these stocks. And this is very important if we want to have informed management actions, plans, and decisions. And also, this is very important in conservation because, as all of us know, resources in conservation are often limited, and we need to make the most of them. And this is just one example of the tiger shark in the Atlantic and the Eastern Tropical Pacific, but if you look for genetics and sharks in Google, you will find a lot of information already available. Genetics have also been important to unveil cryptic diversity, and that is the case of the North Carolina hammerhead shark, Sphyrna gilberti. And this is just a proof of how other lines of evidence alone, for example, morphology, are sometimes not strong enough to provide some information that genetic can give. We have used molecular tools for the identification of shark species involved in predation. This is important in fisheries, so when sharks are feeding on commercially important species, but also for those species that interact directly with humans. It does not stop there. In the forensics field, we have used genetics not only to provide information, but to develop some more applied tools, so identification of species. We now can go to a market, a supermarket, or to a landing port, grab a piece of a shark or of a ray. It could be a clip of a fin or a piece of meat, and identify the species quite easily in most of the cases. We know which gene we should target, we know what the procedure is, and most importantly, we have these massive amounts of data that are freely available in some repositories like GeneBank, where we can compare our sample and identify the correct species. And more recently, we have been using genetics also for tackling illegal trade, right? We just, before the coffee break, had some interesting talks specifically for Greece and Italy on how we go and sample pieces of meat in markets or supermarkets, and we don't always are buying what we are told we are buying, right? Sometimes they are all labeled under a single name, but in fact we are what it's even more concerning is that sometimes, most of the time, these species are endangered as well. And this is just the last, one of the last cases by Diego Cardeñosa, which analyze or look for shark species in pet food and beauty care products. And yeah, surprisingly, or maybe not so surprisingly, there was a big amount of sharks in these products. Now, despite of the importance that genetics have had in elasmo brand research, and as we continue developing molecular tools, we are more and more aware of its limitations. Traditionally, mitochondrial DNA markers or genes have been used to study the boundaries between populations and between species. And we have already identified a couple of these markers that have been very useful. However, there are some cases, specifically for elasmo branch, for which the mitochondrial DNA has a very low evolutionary rate, where these markers are not good enough to tell us these differences between populations or species. And this is the case of the Glyphis glyphis, an endangered species in northern Australia, where after several years of using single gene approaches or a combination of few genes or few regions, there was still not possible to identify population structure. However, in a couple of years ago, they sequenced the entire mitochondrial and combined these with genomic data, SNP data, or single nucleotide polymorphisms, which are also one of the most commonly used genomic data sets. And they were finally able to find a structure. So the authors here recommended that this might be the case for a lot of species. So it's not that there's no structure, it's just that probably we're not having or handling enough information to see that structure. And this is another case, the one of the Galapagos and the Dusky shark. This is not at the population level, but at the species level. So using single gene approaches again, using the ND2 and the control region, it was not possible to tell them apart. So they cluster always together, Carcharodonus obscurus and Galapagensis, till the point that it was even suggested that they might be a single species and that there might be a different morphotype rather than different species. I will talk about this specific case a little bit later as well. So when we move from genes to genomes, we're basically shifting in a scale from using tens to hundreds of markers to using thousands of markers, if not the entire genome. As a result, we're having a more finer resolution at all the levels at which we use species, population, individuals. We have, because we have this amount of data, we have the potential to investigate the effects of selection, local adaptation, gene expression, and epigenetic responses as well. So one by one, at the species level, I'm going to talk about some specific cases for species ID and hybridization. And the first, I said at the beginning, genetics had been an important tool to tell species ID in most of the cases, but not in all of them. Some cases are a bit more complex, and that's specifically when the species are recently diverged. For example, in the case of Carcharinus limbatus and Carcharinus tilstonii, these two black tip sharks are distributed in the eastern coast of Australia. And while the mitochondrial DNA provides accurate information to identify each of the parental species, is not enough to find the hybrids. We know they're hybridizing. They have overlapping distributions. They both prefer coastal areas. One of the main differences between the two of them is the number of vertebrae, and also that one prefers shallower waters versus the other one that prefers more to spend time on the depth. But it was only when using a combination of mitochondrial microsatellites and SNP data when it was possible to identify up to second generation backcrosses of hybrids between these two species. Something similar occurs between two hammerhead species, the Sphyrna lewini and Sphyrna gilberti in the Atlantic. So again, mitochondrial genetics can provide species ID, but not information about backcrosses and hybrids. And the third case, retaking the Galapagensis obscurus case, I said mitochondria is not even good enough to provide information at the species level in this case. So when we use genomic data, we are capable to distinguish the two species and also to detect these hybrids. And here I'm up showing the distribution of these hybrids. So one thing that's very clear from here is that the F1s are distributed sort of in the middle of the eastern tropical Pacific. The backcrosses towards obscurus, so the ones sharing more genetic material with the obscurus, are in the north, and the ones sharing more genetic material with Galapagensis make us think that it might be that hybridization is occurring somewhere in the center or north of the eastern tropical Pacifics, and the hybrids are the ones moving. But this is something we need to investigate a little bit further. But why is this important for conservation? Well, in order to conserve something or to come up with appropriate management plans, we need to understand the status of populations and the status of species as well. And if we keep having error in landing data because morphology is not good enough or traditional genetic methods are not good enough to identify the species, then we need these sort of studies to understand how good or how bad are these populations and species at the moment. Now moving to the population level, genomic data has been providing a lot of important information about connectivity and movement patterns, both historical and contemporary, a much finer resolution in stock structure analysis, information about inbreeding depression and abundance estimates. So while traditional genetic markers have been very important to tell us about the historical connectivity patterns between populations, we are still struggling to identify these contemporary patterns. And this was the first study to identify or to demonstrate contemporary male-based dispersal in the Glyphis glyphis in Northern Australia. So there are a few things that are very interesting about this study. One is that they were finding these contemporary patterns. The other one is that they did this based on sampling juveniles only and investigating the kinship between the juveniles. So they were able to provide information about the adult movement based on these juveniles. What they found is that, they sampled in 3 riverine systems in the Northern Australia. They found first that the juveniles were not moving between these riverine systems, at least not within a 6-year period, that the females are most likely popping on the same riverine system every time, and therefore that are the males that are moving between these breeding areas. So this is important because they are not only providing information about the dynamics of the juveniles, but also about the adults. So perhaps this can provide information on how adults are moving in this area, and in some areas that's very important because we need to create corridors to protect the sharks, the adult sharks. Another case at the population level is the one of the Galapagos shark in the Galapagos Islands. In this case, genetic mitochondrial DNA was not able to give us much differentiation, but SNP data did. A bit impressive because we're talking about a very small archipelago. So the islands, these islands here are no more than 50 kilometers apart one from another. And we know Galapagos shark has an enormous potential to move. So finding structure at this small geographic level, it's very, very important. And it allows us to provide information to the relevant authorities to take this into account when they're coming with management plans. The other application is estimation of abundance. And I want to refer here to this methodology, closed-skin mark recapture, or CKMR, which is basically a genetic mark recapture technique. So instead of using physical tags, we're using genetic tags. And the recaptures are of the individual's relatives rather than of the individual itself. And it's a very new, or a relatively new technique that's only been possible due to all the advances in the molecular technologies in the past 5 to 10 years. It still has some methodological challenges, but as we keep using it in more species and applying it to more species, it's evolving quite well. So it's very good because it allows us to estimate the size of adult population based only on juvenile data, if that's all we have. It can tell us how much are the adults contributing to the offspring, and it can even give us some information regarding the population trend and the mortality rate of the adults. So it's based on the fact that each juvenile or each individual can tag its two parents. And it has, as I said, some advantages. It does not necessarily require to be sampled from live animals, but you can also get the information from landing ports, from markers or supermarkers. It has two ways in which it can be developed. One is if you have access to juvenile and adults. So in this case, you sample a bunch of juveniles, sample a bunch of adults, and then compare the DNA profile to see how many of these adults are the parent of how many of these juveniles. So you find the parent-offspring pairs, and from that, mathematically, you can calculate or estimate the total adult population. The other option is if you have access to juveniles only, which can often be the case for sharks where we have access to nursery areas, but not so easily to adult congregations. So in this case, we sample a bunch of juveniles. We do the DNA profile of all these juveniles and see how many of these are half siblings. So we find the half sibling pairs, and we can estimate the breeding population of adults. Another advantage of this is that there's no more tag reporting bias, which is one of the main problems in traditional mark recapture methods, and that there's no physical recapture needed, so we can do this all in the lab. It has been applied for great white sharks in Australia, and in this case there are two populations of great whites. One in the eastern, where most of the individuals that are found are juveniles, and one in the southern western area, which is composed by subadults and adults. So in the case of the eastern population, the mortality rate, survival rate, and the population estimate has been done quite extensively using acoustic tagging. But that's not the case of the southern western population. So in the case of the southern western, closed-skin mark recapture was used to estimate the mortality rate of adults. And it was used for both populations to estimate the population of adults. So in the case of the eastern, and based on half sibling pairs, it was estimated on almost 5,500 individuals. And in the case of the southwestern, and based on parent offspring, was about 1,500. This is important in species like the great white because historically there is a record of negative interactions with humans, and there's a lot of debate regarding policies for management and how to reduce the risk or the mitigation of shark attacks. So in order to come up with these policies and to come up with appropriate management, we need to first understand what is the population estimate and what the trend of this population is. And this close-skin mark recapture technique has been used for other sharks, has been used for the gray nurse shark, and we are now starting to use it for the black-tip Carcharinus limbatus in Galapagos as well. At the individual level, well, one of the most obvious things after we go and are capable to tell the species from a piece of a shark will be where does it come from. And this has not been widely explored yet for elasmobranch, and I'm not going to talk about that in detail, but what I'll tell you is that we're starting to analyze this for Galapagos sharks in the Galapagos. And we can do this because for this particular species, we have already developed a big genomic resources data set. And also because for this particular species, there's a very strong particular signature in the Galapagos Islands. So we might not be able, next time we go and catch a part of a Galapagos shark, we might not be able to tell if it comes from Colombia or from Peru or from Mexico, but we will be able to tell if it comes from the Galapagos or not, which is already a good first step towards the development of these tools for other species, but also to start being able to say exactly where does it come from. And the implications here are very obvious. So I'm going to spend a bit more time into talking about the sex ID and the duplicate ID. So far, how we determine the sex for sharks is basically depending on whether we see the claspers or not, right? We go to the field, we check if it has claspers, we record it as a male or as a female if it doesn't have, but there are a number of factors that can interfere or can come in the way when we're doing that. First, if we're sampling under not ideal conditions, if the visibility is bad and we're under the water, it might be difficult to check for the claspers. It will depend on the size of the individual as well and on the position of the observer respect to the animal. So we don't want to take the risk there. And it's even worse when we're talking not about full individuals, but about individual parts from fins or from meat. And we now have access to these big data sets. We can have access to a lot of information and actually use genomics to determine the information, even if it's from just a part of the body. How we do that? Well, specifically for a species with heterogametic sex determination systems, like we humans are XY, what happens is we find that the markers that are associated to the male chromosome, so to the Y chromosome, which will be only present in males and will be absent in females. But from that, we can target and resequence these specific markers and develop some short PCR assays to determine if it's a male or a female. Now the question is, can we do this in sharks? And the answer is yes, at least for 20 sex determination system, which is also XY. So we have found the Y-linked markers, and in some cases we have already, like in all 20 species, we found these markers that are only present in males. And for some, we're starting to develop the PCR assay. So here are some preliminary results. These are for the great white shark, for which we were lucky enough to have a reference genome, so we were able to target not only these SNP markers, but longer sequences. And as you can see, it will be hopefully as easy as running a PCR to detect if it was a male or a female. So I don't know for you guys, for me it's pretty exciting. And another very obvious application of this genomic information, of course, is the identification of duplicates in our sample. So for all of us who have been in the field, sometimes these are the conditions, if we're lucky sometimes, under which we are sampling. I swear this is not a rock, this is a marble ray. And as you can see, if we take the sample from this individual and we keep swimming, it might be very, very difficult to know if we have already sampled that animal or not. Also, when we're going and working from samples in markets or in ports, it's difficult because sometimes we find only the parts, and you don't know how many of these fins belong to the same animal or not. As well, some species do change their coloration pattern, which is often used as an individual ID. They will change in color pattern as they grow old. And in some cases, for example, in the Pacific spotted eagle ray in the Galapagos, we have found that the coloration pattern changes within 2 months. So that's not, that's very important. It's not good enough unless we have an external tag. We might not be able to say if it was the same individual or not. And sometimes there are some marks that the individuals might have on their fins that are already there and that can get confused with the mark we leave when we take the fin clip. So we now don't, this is one less thing to worry, as we can run some tests in the lab and bioinformatically discard all these duplicates. So the take-home message here is genomic information and the genomics toolbox should be a common practice in conservation. It comes with a lot of advantages. As I said, simpler sampling methods. We only require tissue or DNA to respond a lot of questions. In terms of more applied conservation, estimating abundance, it's crucial to assess the efficiency of management actions. Yet, this is one of the parameters that's still very difficult to estimate. So having mechanisms like closed-skin mark recapture are very important for us, and we should start using them. And the statistical power and the improved resolution with genomics, it's also really, really good. Some recommendations to finish the presentation. Whenever possible, keep collecting tissue samples on a regular basis. From experience, all the samples that have been collecting in Galapagos for years, even before I came back to work there, are now going to be of great use to respond these very important questions for management there. Keep the associated data when possible and collaborate there. We are lucky now, we have a lot of platforms available for us to share samples, to say to the Elasmo branch world, I have these samples, or to request samples and start moving from local to regional and global assessments. Thank you very much. Acknowledgement to all my collaborators and the people who is working and whose data I have presented here for you.
Thank you very much, Diana, for this very interesting presentation. And any question? There.
Hi. How many SNPs did you use for the Galapagos shark study? For the stock assessment, how many SNPs did you use?
How many SNPs? For the provenance we're working on? For the population structure, we had around 10,000 SNPs, and then we are exploring with 200 SNPs that are very informative for the particular Galapagos population.
Other questions?
I only wanted to know if you were able to detect hermaphroditism in your samples, and if you can, the degree of its appearance.
No, this is something that not yet.
Any other question there?
And thanks for the nice talk. I was wondering if you how much sampling effort do we have to make for the closed-skin mark recapture?
Yes.
I'm guessing that you'll have to sample a lot.
Yes, you have to sample a lot. So There are a few requirements you need in advance before trying even with closed-skin mark recapture. One of that is have at least a rough estimate of the total population, because you calculate the minimum you will require on sampling. So that's a good news and bad news. So it's good news. I don't know if I should call it good news, because it's not good that populations of sharks are often small, but it helps because it's an indicative that our sampling it's not going to be on the millions.
Have to adjust according
Yes.
to the estimated population.
Yes.
Thank you.
Any other question there?
Do you know of any examples of being able to identify individuals?
Like a specific individual.
We're working on white sharks in California, trying from eDNA to tell individuals. I was wondering if you've thought about that in your work.
Like personally, no. I know there is like some effort to try to do that in Costa Rica as well with the tiger sharks, trying to aim for like the specific shark, and there's some attacks, but I'm not aware of a successful case already.
Any other question? If not, thank you very much, Diana.
Thank you.
Francisco Colloca, please. So using local ecological knowledge to reconstruct trends in elasmobranch population in the Strait of Sicily.
So thanks, chairs. Good morning, everyone. So I'm going to talk about a study that we are carrying out in the Strait of Sicily. Is about using local ecological knowledge from fishers to try to reconstruct population trends in this region. And as already said this morning, the Mediterranean is considered as a kind of biodiversity hotspot for Chondrichthyes. But at the same time, it's a world area with the highest proportion of threatened species. We have 88 species now described in this region. And someone talked about as an hotspot of extinction risk. So we have a lot of conservation issues in this region. And the other big issues that we have is that it's difficult to implement management measures and a good management strategies for this group of animals, because we don't have much knowledge for many species. This is, for instance, the IUCN, the last IUCN elasmobranchs assessment for Italian waters. So you see that almost 50% of the species considered are data deficient. So we have quite strong knowledge gaps that need to be filled, and using the local ecological knowledge of fishers can be an alternative way to gather data, because we need to gather data, we need to improve our knowledge about many of the species that are currently endangered in this region. So we focused on the Strait of Sicily. Why? First of all, because our lab was on the southern coast of Sicily, in Mazzara del Vallo. And the second reason is because the Strait of Sicily is one of the most important areas for elasmobranchs in the Mediterranean, both in terms of biodiversity and species abundance. And there are important catches of different species of elasmobranchs from different fisheries, either on the Italian side and the African side. For instance, in the picture here, you can see a photo of landings of smooth hounds during 80s. That is a common, still common landings now in the region. But the problem here is that we have big lack of knowledge for population trends for the main species, and most of the information is limited to the last 25, 30 years, which is the period when started more standardized data collection in the Mediterranean. And so our objective were to investigate the impact of fisheries, troll first of all, on the last Marine community of the Strait of Sicily, as perceived by fishers, since the end of the Second World War. And the second one was to identify the areas of very high CPUE, meaning the areas where there are concentration, or there were concentration of individuals of the different species that can be regarded as areas for mating, parturition, feeding, or nurseries. So the methods. We carried out an interview surveys in 2016, 2017 in Mazzara del Vallo Harbour. Mazzara del Vallo was, and it is, still it is, the biggest troll harbour in the Mediterranean Sea. It hosts nowadays a fleet of about 90 big trawlers fishing everywhere in the central and eastern Mediterranean. And you see from the graph the history of the fishery. Sorry. You have here, just after the Second World War, you see that the numbers of, of fishing fleet above 14 meters length was really low. There was a quite rapid increase during the 50s and the 60s, and the fleet picked up at the end of 80s with almost 290 big fishing vessels. And you can imagine that the fishing capacity of this fleet has increased exponentially, considering that a small vessel were replaced by big trawlers. And so it's extremely important to gather and recover information from the initial period of the development of an industrial fisheries here in this area. And so for doing that, we need to recover the memories of the old captains, the generation of fishers that was fishing in this period. And because it's sad to say, but these fishermen are old and are going to pass away. This is life, unfortunately. But we really need these memories because are so important to define and establish a kind of baseline for species diversity and abundance. So what we did is basically to replicate a methodology that was already done during a ProIU project, EvoMed, that was carried out some years ago in different sectors of the Mediterranean. The scope of that project was the same, recovering local ecological knowledge from fishers, Mediterranean fishers. And what we did is to interview 28 old fishers, retired fishers from the Mazzara troll fleet. Most of them were from the trawlers, but there were also some artisanal fishers. And you see from the distribution of the ages that most of these fishers were over 70 years old. So they witnessed the catches during the early development stages of the trawling in the Strait of Sicily. So the interview survey covered the period from 45 to 40, to the present. So divided in three main periods, segments, from 40 to 60, 61, 80, 81, 2000. And the fishermen were asked to rate the catches in each period compared with the present. So what we asked is, tell us if this species is abundant as in the past, is less abundant, if it is more abundant, how much is more abundant, 2 times, 3 times. And we asked also to the fishers, remember the landings, because they have very good and precise memories about these exceptional or unusual catches for the different species. So we show them a map like this, showing the different position, the different fishing ground of the Mazzara fleet, asking them, please tell us where did you got that catch. And we focused on a bunch of 18 different species, which can be considered as the, let's say, the most important bycatch species for trawling. They are both rays, sharks, most of them are demersal, of course, exploited or bycatched on the continental shelf and also on the continental slope. And what we get is something like that. This, I cannot show you all the full results, but just some example for some species. For instance, this is the case of the smooth hounds. We have 3 species in the Mediterranean. In this case, we put together Mustelus mustelus as Mustelus punctulatus because Fisher, they do not recognize the two species. They put together in mixed boxes. And the graph here show the distribution of the frequency of the responses, and you see that the perception for these porous mutants is a decline. 2 minutes, okay. Is a decline that is 2 times, 3 times. And you see from the map the position of the areas where the eigensketch have been obtained. And you see that the catch that they obtained was up to 3 tons per day, so it was really amazing. And this is the smooth hammerhead. It was common in this region until the 60s and the 70s, both on the Italian side and the African side. Now it's disappeared since the 80s, completely disappeared. And the last catches was recorded in the 80s in the Mammellone, so-called fishing ground. Mammellone means in English big teeth. And fishers refer to winter aggregation during southeast to Lampedusa Island, and the catch was up to 60 individuals per day. So I will skip this because I don't have time. Just say something on squatina. Squatina also disappeared from the region. It's considered as extincted, basically, by the fishers. And, but it was exploited by specific fisheries, artisanal fishery during the 40s and the 60s. And during the 80s, the catch was up to 150, 200 individuals in a single hole. So this is the global picture for the species that we investigated. You see that the perception of the fisheries that all the species declined at least 2 times, but in most of the cases 3 times. Some of them are absent now from the region. The majority are rare. Few of them are still common. So, conclusion. What we need is to gather more information. We are expanding the service to the eastern side of the Sicily. It will be nice also to include Tunisia. We will see what we can do. The current status of elasmobranch species tell us that almost all the species are considered as strongly declined during the last decades. We need special conservation action on an urgent basis. It's time to take some relevant action. We cannot just limit to reduce the catch of some species. Probably we should start thinking seriously to a troll catch ban or a bycatch ban for most of the species. Or sorry, a landing ban for most of the species. Okay, I'm going to finish. So let me say co-authors, of course, but especially to the fishers of Mazzara del Vallo. We have 2 associations, our retired fishermen, the association of Casa del Pescatore, association that collaborated for the studies. Just to finish, a positive message to conclude. This is a photo of an angler shark that was caught with a ROV in the Aceste seamounts in October 2018, which means that probably some of these species is still present in some refugia. And what we should start thinking about is to study this refugia, also in this case taking some conservation measure, investigate how much these refugia are connected between them. So, thank you very much for your attention.
Thank you, Francesco. Any question?
Do you ever have problems with like fishermen dying because it wouldn't benefit them to have a ban on like the trawl boats because they'd lose out on money?
I don't think, because they are retired, so basically they're not much interested in what's going to happen in the fishery. But so I don't think that they are lying. But what is important is to focus on the maximum catches they observed, because they can be remembered very well by them. If you ask, okay, what is the average? It's something that is really vague and difficult to be quantified.
Okay, thank you, Francesco.
Okay.
So we don't have time for other questions. Thank you. And the next talk by Giulia Prato, Action Towards Collaboration for an Improved Bycatch Management in the Mediterranean, Conservation, Communication and Citizen Science. Thank you.
Okay. Good morning. Thank you very much. I'm working for WWF in Italy, and I'm going to try to give you a quick overview of the first pilot project we are having as WWF in the Mediterranean that is obviously embedded in a wider Mediterranean strategy for sharks conservation. So we know, as it was mentioned several times today, that the Mediterranean is one of the most dangerous seas in the world for sharks, with more of half of the species threatened. And just before the summer, we published the report Sharks in Crisis that was mentioned before, thanks for that, where we were assessing a little bit the situation, the current situations in the Med, and what are the main threats to sharks here, and we also put forward some policy recommendations. The report was developed with the support of a lot of people in this room, actually. And we know that bycatch is the main threat for sharks in the Med. So Safe Sharks is, as I said, the first pilot project that is we are dealing with as WWF with this topic, and it's in collaboration with the COISPA and INCA, which is a small NGO in Albania. The project is focused in the southern Adriatic. It started in 2018, and we are now towards the end of the second year. Its main objective is to promote and improve management of bycatch in Italy and Albania, and through four main lines of action. So improving the knowledge about bycatch phenomenon in the Southern Adriatic through stakeholder engagement and citizen science, develop solutions to increase post-release survival rates of sharks. So we are using electronic tagging to assess post-release survival rates, focusing on blue sharks. Then we are also working on the market to assess traceability of the supply chain and seafood fraud risk. And then obviously we work within the regional strategy with advocacy actions and awareness raising on the issue of bycatch. The last, the first summer of the project, we completed this questionnaire survey involving the fishermen of the longline fleet targeting swordfish in Monopoly, which is the main fleet in the Adriatic targeting swordfish. We did the same interview in Albania, I'll show you later. And we've seen we managed to cover all of the fishermen. So here the data is not updated with the whole of the fleet, which is 20 fishermen. Here we have only 15. We've seen that in this activity, there are more or less 14 species that are often subject to bycatch. The more common ones are blue shark and the devil ray. And among these species, several are protected under the Annex 2 of the Barcelona Convention, and they are under protection under the GFCM recommendation as well. So they cannot be retained on board or landed. So we have devil rays, makos, treasure sharks, poor beagles as well that are sometimes also bycaught. And among the main species that are discarded, we have the devil rays, but also the blue sharks. Generally, they discard the smaller ones, so under the 10 kilos. According to the fishermen, when they release the sharks, these are generally alive. And overall, they stated that it was clear they had a low knowledge of the current legislations, so they don't always know what they can land and what not. And sometimes the information they gather, it comes from their own retailers, those that buy the fish from them. And sometimes the information is not correct. In Albania, the situation was similar. We did interviews in 3 main ports. We didn't target a specific fleet here. We targeted trawlers, hook and lines, and trammel nets, fishers. The trawlers were the ones with the highest diversity of catches. Also here, several protected species are caught as bycatch. And actually, in some cases, they're also landed. So they were stating that they would land as well sometimes devil rays, as well as makos. They also mentioned white shark. Again, here in Albania, the identification capacity on species seemed to be quite low, as well as the knowledge on legislations. We also tried, as before Francesco did, to collect some of the fishers' memoirs on trends that they have seen. And apparently they've seen a decline in the catches of angel sharks, guitar fishes, and hammerheads. But also surprisingly, especially in Monopoly, they were stating in the past 2-3 years to have seen more big-eye treasure sharks, which they have never seen before, as they said. Concerning the perception on bycatch, they luckily, more than 60% of them, they don't think they're important for their fishery because of the low economic value. And 40% of them think that sharks are actually important for the environment, which is pretty good. And when we were trying to understand whether bycatch is an issue for them, it was nice to see that it is actually, because for them it is a loss of time, the time lost to handle the animal, release it, or putting on board. It can cause damage to the gear. It can have an impact on the security of the crew. And also they risk sanctions because, as mentioned, they don't know what they can land and what not. All of this for a very low economic value. So considering all this information we gathered talking with them, we decided to organize a few workshops, also with the collaboration with Carlotta. We organized a workshop in Chioggia and another one in Monopoly, with some trainings on species identification, current legislation, and we invited to these workshops like fishermen, but also retailers and Coast Guard, and there were all the stakeholders actually were really stating that there is a high need for this kind of activities and more information. This summer, we also started the tagging program. So we're using one month electronic tags, so for assessing the post-release survival rates. We deployed 7 of these tags for the moment, and we are aiming to achieve, we have 15 to deploy in total. We are also going to deploy one year round electronic tag to follow just one individual. And at the same time, we are working with the fishermen on best handling protocols to raise their awareness, on the need to properly handle the animal before releasing it. As said before, we are also assessing the risk of seafood fraud on the local markets. So we're concentrating in the Apulia region around Monopoly, gathering samples from fresh fish shops, also shops specialized in frozen fish, supermarkets, etc. And we're gonna assess the risk with DNA analysis. And we're also assessing the supply chain of 2 main retailers, local retailers that buy products from the Monopoly fleet to provide recommendations to them to improve the traceability of their supply chain. And then we are also, while during developing the report, but also while working with the fishermen, we realized how much the data is scattered across the Mediterranean, how many separated different little data sets are available and hard to access. And this is also this is also demonstration that countries are not really complying in terms of data reporting with the complex range of legislations that we have in the Mediterranean. But at the same time, we saw that there are a lot of citizen science initiatives that have spread in the Med in the last years, based on apps to collect data or initiatives that collect data on sharks through social media. So we decided that it would have been good to try to bring together all these initiatives, to talk to each other and to see whether there is a way that this kind of information could be of support to bycatch management. And this is why we organized a workshop on citizen science here a few days ago on the 15th. It was really great to see that there was a really high participation, so thanks for all that came in. And during the workshop, we discussed 4 main topics. After seeing all the, let's say, the initiatives that are existing, we discussed 4 main topics, which were the strengths and weaknesses of the presented tools. We analyzed the point of view of the user in terms of user needs, and with user we mean the professional fishermen mainly. So what are the needs in terms of applications or tools to report and to comply with the current legislation, but also in terms of usability. Are the produced tools actually usable from their side? We also discussed about how to capitalize on the existing initiatives to feed into a regional data framework, if that would be possible. And we discussed about sensitive data and legal aspects related to this. And as let's say, these are the first set of key points that came out from this workshop. First of all, there was a general feeling that there is a need, there is a need to really start from the data and try to get some standardization of at least a few key data that could be useful for conservation and management that everybody could collect, while being careful and not falling in the standardization trap that we develop a new standard actually, and there are already many. Then there is a general agreement that the different initiatives should talk more to each other, and there were some proposals about one potential platform that could host all of these initiatives, through which we could have access to these initiatives, maybe even to the data through a common data policy, potentially. And then a main topic of discussion was the real strong need to involve the user, the fishermen, since the beginning in the development of these tools. So the co-creation approaches are fundamental. And it is also to really properly assess the user needs and the usability of the tools. And it is also really important to give back to the users, so potentially with electronic education tools, such as simple species identification guides, or just really having them involved in the process. And here you can see one of the fishers of Monopoly with the Shark Angels t-shirt. And he also discussed whether these apps could be in the future of help for the fishers in compliance, in respecting the current regulations. So just very quickly, we're going to produce a summary report of this workshop, and we're going to share it for validation with the participants of the workshop. And actually, we're going to share it with those who signed the paper. So if you didn't, please come over to me and Simone, and we'll try, and we will share it also with you. Thank you very much.
Julia? Any question or comments? Okay, just a comment. I found very interesting the workshop too. So
Thank you.
thanks for organizing it. And so we can move on now.
Armel Jung. Okay. Data collection and conservation campaign for sawfishes Priside in Indonesia.
Hello everyone. I will try to raise your attention more before lunch. So my name is Armel Jung. I'm gonna talk here to you today on behalf of my team about a project named Indonesia, data collection and conservation campaign that is carrying on in Indonesia. Our team is composed of Professor George Burgess, who I don't know if we need to introduce him, but he has dedicated most of his career among other species, but on sawfishes, and is newly retired from Florida University and Museum. And Dr. Damadi, which is in charge of the Elasmobranch research program in Fishery Research Center of Jakarta. For the ones that are not familiar with the species, a few information on a general scale. There's 5 species of rays that are inhabit coastal and marine brackish water in most of the tropical zones, but also in estuary and lake for some of them. They can reach up to 7 meters, and they're found in an average depth of 10 meters in mangrove or sandy bottoms. In Indonesia, we can find 4 of the 5 species world around. The narrow sawfish and the dwarf sawfish that are listed endangered, and the large tooth and the green sawfish that are listed as critically endangered. All the species are also included in the CITES Appendix 1 since 2006. Indonesia is located in Southeast Asia. It's one of the biggest countries there. It's composed of more than 70,000 islands. It's a very complex archipelago composed of very small islands. It is considered world around as one of the top biodiversity in the world, top 3. But at the same time, it's also the top 4 for human population. Sawfishes and Indonesian people are linked and have a strong relationship for a long time. As you can see here, some hard craft I would have loved to offer for auction yesterday, but they belongs to Paris Museum. I'm sure we had a good price for it. And these pictures from our research work in Papua. This has been very well developed by an ethnographical study by Matthew McDevitt. Indonesia has protected sawfishes for a long time. They are included in the law since 1999, and it is absolutely prohibited to catch them. Despite of that, a lot of bycatch still occur, and that's why we started this project following the global action for sawfishes developed by IUCN Working Group in 2012. To do so, we created a network of focal points that are referent people in each province of Indonesia, and that are working with a team of enumerators that go on each landing places to collect some data. We also had a lot of awareness raising through a specific poster that has been designed by local people, which also included a little code of conduct and contact of the people in case of bycatch. The project consists also a lot in capacity building, so we dedicate a lot of time in training. First of all, training the focal point in terms of biological knowledge and data collection, but also training them in order to perform good inquiries and become, at their turn, trainers for their enumerators, and also doing good sensitization with fishermen. We succeed in performing up to 1,000 inquiries in the different provinces and being in touch with 8 shark fin traders. We also succeed in getting access to the national statistic information from the country, which is not always easy. But here is the figure we had. Honestly, George and I were a bit doubtful when we get this figure. But after the 2 years' work that has been done, we had to conclude that at least 5,000 sawfishes are caught every year in Indonesia. And this was a combination on all the information that we gathered from the seizure that have been done, from all the field work, from all the control in the landing places that has been done. These landing controls, to give you an idea, this is how a sawfish parcel is prepared for export to Hong Kong or other Asian market. There's a labeling on it here mentioning Radiorhynchus australiae, which belongs to the bottlenose wetfish. But when you open the parcel, you did realize that, in fact, in the middle of that, you have a narrow sawfish inside. Our focal point are trained to recognize the different shape of a fin, but you can imagine it's not always easy when you have to fight with mountains of fins on the floor. However, we gather all the information from the inquiries, and we were able to start to build a distribution of the sawfishes from the recent years. And we confirm that the sawfishes are still very present all over Indonesia, with a special hotspots that we found in the extreme east, in the Arafura Seas, close to Papua New Guinea, and in the northern of Sulawesi. It was also many times mentioned that this area included the nursery for narrow sawfish and pristis, the large tooth sawfish. However, up to now, we haven't been able to confirm the presence of a dwarf sawfish that is present in Australia, very close to this area. So it would have been interesting to find out more about this. About awareness raising, we have also been working a lot with WhatsApp, especially, that have been very useful for communication between fishermen and focal points. You can see here, for example, a screenshot of one of a focal point in Papua, Gunawan, with his kids behind, and then just receiving information straightforward from the fisherman that is giving position of last catch. Also, just to show you one of a nice video that was sent by a fisherman, also through WhatsApp. This catch and release has been sent by people in the middle of the Arafura Sea that were able to get informed after a awareness raising campaign that was done by the team, and then they decided to release the sawfish alive, to film themselves, and to post it on the website from the project. Anyhow, it's a bit tricky to work in these areas, and there's a lot of caveats, and the national context for research is quite complex, either for international researchers but also for national ones, due to the complexity of the administration there. And the size of the country is also a limit, for sure. Anyhow, these first results have proven the positive action that we can have by working straightforward with the fishermen. It has been said previously by my colleague before, but there is a strong effect by working jointly with the communities. And we would love to be able to continue this work and to do more analysis of what has been done, and especially with DNA. And also mention that yesterday it was International Sawfish Day. I wish every day would be Sawfish Day. You have to know that in Europe every day rostrum are sold. So there is another project we carry on with an NGO which is called Merci, where we are trying to work about illegal trade of CITES species, and I invite all of you who are interested in that to join this project. I thank you for your attention. Thank all the people from the team working hard in Indonesia, the Save Our Seas grant to fund this 2 years project, and the fabulous organization team for this year. Thank you.
Thank you, Armel, for this speech. Any question, please? There is time for questions.
Maybe today, despite Francesco in the lab, is the first day I realized that this may be the prime candidate for an anti-poaching tag because of the high recapture from the trade. So have you thought about using electronic technology to really start to get right to the heart of this problem?
You mean using satellite tech to better... I can't hear you very well.
I think that you could chip this fish and really make a difference to pinpoint where your problems are. And it might be the prime candidate of all sharks on the planet.
Yeah, for sure. Yeah. Thank you.
No more questions? Okay, thank you, Armel.
Thank you.
Thank you very much.
So the next talk will be by Melanie Villate Moreno, A Blank Area on the Map, Molecular Identification and Shark Population Dynamics, Implication for Conservation Based on Artisanal Fishing. Thank you.
Hey, good morning, everybody. Thank you very much for being here. way of locating you a little bit. So we're talking, where's the, this is the pointer? Okay. Yes. So we are talking about the Pacific coast of Colombia in a region that's called Chocó, so this area, and this region is in the eastern tropical Pacific. This is an area well known for having a high productivity, having high biodiversity, and this eastern tropical Pacific is a well-known corridor for different organisms. So we have turtles, dolphins, sharks, whales. And the coast is characterized by having estuarine ecosystem and mangroves, which can serve as nursery areas for different species. So in this area of Colombia, we have two different type of fisheries. We have the industrial fisheries, which are known for having a higher impact on the environment and a high amount of bycatch, but we also have the artisanal fisheries. And the effect of the artisanal fisheries in the species is not so well known in this area. So they catch the commercial species, but they also have a lot of bycatch. And why are we studying this area? Why do we think this is important? So in this region of Colombia, in Chocó, we have a marine, a national natural park called Utría, and this represents 5% of the total surface area of the region. Only from this 5%, it is known that it provides more than 17% of the catches of the region. And if this is happening only in this 5%, what is happening in the other 95%? We have no monitoring program, we have no data collection, and we don't know anything about the taxonomic analysis, so we don't know which species we have in this place. And then we get to the sharks. So as I was telling you, the fishermen go, they fish for the commercial fish species that they're interested, but they also bring a lot of catch as bycatch. In this case, what we wanted to do was use a molecular approach. And we're using this because, as Diana was explaining before in her talk, in this case, the fishermen come to shore with the sharks. They have no fins, no head, and no tail, and an anatomical or morphological identification is very difficult. So what we use is we gather a piece of tissue sample and then we analyze it genetically to know the species. We use, in this case, mitochondrial gene, the ND2. So for showing a little bit the results, this was surprising for me that the shark that was, we collect more than 700 samples. I'm just showing like the first analysis that we did. The first shark that was being fished the most was the hammerhead shark, and it is more or less the 33% of the samples, and this shark is known as being endangered worldwide. So hammerheads are top predators, they have cast-selected life histories, so this means that for the populations, because of these ecological characteristics, it is difficult to recover under exploitation events. The second species that was being fished the most was a species of Mustelus, but this one is a mesopredator. They grow faster, they get to the reproductive stage faster and have more offspring compared to the hammerheads. Then we have another species of Mustelus, and then we have the silky sharks with 10%, which are also vulnerable, are also top predators, and they also have cast-selected life history. In total, we have 10 species that we have found so far, but these ones are the ones who represent less than 10% of the catches. So, we have the results from the species. We know which species are being bycaught, and we wanted to know how many females, males, and if they were adults or juveniles. So what we did is, whenever we got the sharks, we measured them, but as I told you, they have no heads sometimes or no tail sometimes, so we couldn't have the actual measure of the shark. So what we did is that we measured the space between the first and the second dorsal fins, and then make some calculations specific for every species, so we could have the total length of the shark, and we always check if they were females or males. So to explain a little bit what this graph means, in here blue we have females, red we have males, and yellow we have unknown. And unknown means that the sharks came without the pelvic fin, so we couldn't know if they were females or males. And we have two different lines. We have the dotted line, which is the mean of the values, and then the straight line, which is the minimum maturity size for every species. So just to give you a little bit of an example, we have here the different species, and let's take this one. So we have female hammerheads. Then the line here means the minimum maturity size. So everything to the left of the line would mean they are juveniles, and everything to the right would mean they are adults. So in the case of the top predator sharks, we have that all of the samples we obtained were juveniles, which was nice result. So we know we have a nursery area, but that also means that the fishermen are fishing sharks that have not any offspring yet, and this is very worrying for the populations. And in the case of the both species of Mustelus, we have either juveniles and adults, because as they grow faster, even if they are small in size, they could be already adults and have offspring already. And then we wanted to know, okay, we know the females, we know females, males, we know there are juveniles, and we wanted to know how was the population genetic diversity. So we used the same samples, we used the same mitochondrial gene, and we used 2 different programs for the summary statistics and the haplotype network. And what we did here is, first I'm going to talk about the haplotype diversity. So that means we have a species, we have all of the sequences from the different individuals of this species, and each sequence is called an haplotype. So what we want to do is how many haplotypes per species we have. If we find many different haplotypes, we would say this species has a high diversity. If we find a low number of haplotypes, then we will say the diversity is low. So in both cases of the Mustelus species, the diversity was very high in this case, and for both top predators, it was very low. Then we have the nucleotide diversity, and what it means is, so we take again these sequences, these haplotypes, and we want to see in how many places. This would mean if they are different in many places, this nucleotide diversity will be high. If they are just different in one place, then it will be very low. And for all of the species, it was very low. And this could be possibly because the populations can be going under a bottleneck. In this case, we only use the species that represents more than 10% of the catches, because if we have only 1 individual or 2 individuals per species, then the statistical results would not be very valid. And here I wanted to show you a map and the different places where the samples were taken. And what we have here, what we have here is, so the network, the size of the balls represent how many sequences we have of the same type. So most of the sequences were like this green. We have the same sequence. So most of the places are not very variable for the hammerheads. But in this place, we have the most of the diversity. So we could consider this place as an important nursery area to protect in the future. Then just to give you another example, we have the silkies, and what we can see, we have almost no diversity at all. We just have 1 sample which is different from all of the others, and in all of the places we have the same haplotypes. And in the case of the Mustelus, just wanted to show you that they are very diverse, and almost all of the places, the diversity is very high. Just to show you like more or less how it looks. After having these results, what we wanted to do is sharing the results with the community. So if we produce all of the information and keep it at the house, at the library, or in a paper, then the community cannot use this for like actually protecting the species. So we went there, we explained why we did the projects, what sharks were important. This was kind of a poster that would happen if we take sharks out of the environment, and how at the end, so how it would look at the end, how the commercial fish species will be affected, how this will affect local community, and also the food safety of the place. And what happened at the end is that, so Chocó, this area, is a collected territory. It means the land is not owned by different people, but from the community, and this community can make the decision, they can be part of the decision-making process, and then they can decide how to use the resources. So it is very important to communicate the results with them so they can actually be part of the decisions. And just as conclusions and further work, we would say that for both species of Mustelus, they would say they will be more resilient as they have higher genetic diversity. We found potential nursery areas for different species. We will say that the top predators have an haplotype poverty because they were not very diverse. And also, as Diana was saying, we only use this mitochondrial marker here. So for what we're doing now is using SNPs, so to know the story of the different genes. And it will be important to include this kind of information in the programs of conservation of the country in the future. Pancho, thank you.
Thank you very much, Melanie. Any question?
Hi, Melanie. I was wondering if you had any plans to sequence other mitochondrial genes and compare what you found with data that's available for at least 2 of the 3 species that you show, because I know there's mitochondrial DNA sequences available for the Cacarinas, for the Sphyrna, at least. I'm not sure about Mustels, but maybe. So just to put your results into perspective and to try to assess how unique these populations may be or not. So just to be careful about the conservation implications that you're deriving from the data.
Yeah, so we know there are so CO1 from some of the control region from the mitochondrial, and it will be nice either from the mitochondrial genes or microsatellites or SNPs to make a comparison, so we could know in the whole area how is it looking, if they're migrating somewhere else, if the places are connected. But yeah, that will be for the future.
Any other question? Okay, thank you very much.
We are ready for the next talk. Francisco Garzon, baseline assessment of the coastal elasmobranch fauna of eastern Cabo Verde using baited remote underwater video, please.
Hello again. Sorry you have to listen to me once more, but I'll be quick. We'll be at lunch soon, and it's very different today. So I wanted to talk to you about our baseline assessment of the elasmobranch assemblage of Cabo Verde. Cabo Verde is an independent country made of 10 islands, and it's located about 500 kilometers in front of the coast of Senegal. The 10 islands are actually quite different from each other, both in culture, population, and environment. You can find islands that have tall green mountains or harsh rocky landscape, or of course beautiful white beaches with crystal clear waters, which is why Mar Alliance has decided to work there, mainly. The actual reasons for our work and continued effort there are different. Firstly, Cabo Verde has been recognized as a marine biodiversity hotspot, not only in the Eastern Central Atlantic, but on a global scale. We have counted about 55 species of sharks and rays in the country, though we expect that the number will increase, especially when we start looking at the deep waters, which are pretty much unexplored. But at the same time, along the coast of West Africa, data and reports are suggesting that sharks' population there are declining very rapidly. Most stocks are overexploited. And this brings us to a second reason to work in Cabo Verde, which is that although shark population seems to be doing relatively well compared to West Africa here, the increase in population and booming tourism are actually driving the expansion of especially small-scale fisheries. And this is likely to pose a threat to sharks and other fish species here. At the same time, there is no real baseline assessment of the abundance, distribution, and assemblage of shark species and ray species in Cabo Verde. So it is hard to plan conservation or plan management of fisheries without data. So the objective of this work was to create the baseline data needed for the management and conservation planning, and also try to assess the current status of fishing pressure on sharks and historical trends. So to do this, we actually used 2 methods. We used baited remote underwater videos to investigate abundance and diversity of sharks and rays. And we used structure surveys with fishermen to see what they knew, what their traditional ecological knowledge was, and how they felt the marine resources of Cape Verde are doing at the moment. So between 2015 and 2017, we deployed 189 BRUVs, and by we I mean the Mar Alliance team there. I wasn't there to do that, and I'm not too sad I did not have to pull all the BRUVs up from 20 plus meters. But the BRUVs locations were selected to get a homogeneous coverage of the coastal environment of specifically 3 islands, Sal, Boa Vista, and Mayo, going from north to south. We hope to expand the work to other islands too in the future, collaborate with the other NGOs there. And at the same time, we also conducted surveys in all the 3 islands, and questions included background information on the fisherman lifestyle, but also which species of sharks and rays they catch most frequently, where they catch fish and sharks, and their perceived trends in abundance of sharks and fish. So from all our BRUVs, we cited 216 individual sharks and rays, belonging to 15 species that we could identify. The most common are here displayed, were the common smoothhound, the Atlantic weasel shark, which is actually endemic to this region, the round stingray, and the nurse shark. We also had sightings of other large sharks like lemon sharks, spinner sharks, black tips, and tiger sharks, though these were rarer than we had expected. We used sequential max-N to compare abundance of sharks between islands and between regions within the islands. And sequential max-N was calculated by counting the number of individual sharks and rays that we could see in the first 60 minutes of our BRUVs. And as you can see, sequential max-N, or the mean sequential max-N in all the islands, was pretty similar. It looks to be somewhat higher in Mayo, but actually the difference is not statistically significant. More interestingly, though, we were able to identify some hotspots of abundance for sharks and rays in all 3 islands. And these actually overlapped with what fishermen told us were the best spots to find both fish and sharks. Although abundance was pretty similar between the islands, actually diversity and the composition of the shark assemblage was different. We calculated diversity using the Shannon index, and what we found was that in Mayo it seemed to be somewhat lower than the other islands. So we went and looked into why that might be, and we found that actually Mayo was lacking some of the bigger predators, like black tips and tiger sharks. And if we look at the species composition of our sightings in the 3 islands, we can see that in Mayo, the bulk of sightings was actually made up of smoothhounds, weasel sharks, and nurse sharks, which are somewhat the lower trophic level species of all our sightings. We can also see a clear declining trend in carcharodonid abundance, going from the northern islands of Sal down to Mayo, the southernmost island. And the same can be said for big, large benthic rays. And it is somewhat worrying to see the lack of big predators in Mayo, especially given that the islands are pretty well connected. Animals, we have tracked animals moving between them all the time. And the environments in below the sea are actually quite similar between the islands. So it is unlikely that the lack of predators in Mayo is due to natural causes, but could be actually a result of exploitation. So after we got this first picture of the shark assemblage in our 3 islands, we went to look at traditional ecological knowledge to supplement our data. What we found was that, interestingly, all the species we cited in BRUVs were mentioned by fishermen as being either possible or regular catches in their activities. But even more interestingly, they actually mentioned a lot of species that we had not cited in our BRUVs, including the hammerhead, blue sharks, shortfin mako, and sand tiger sharks. In fact, when we looked at what the most frequently caught species were in Sa and Boa Vista, we found somewhat alarmingly that hammerheads were actually at the top of the most commonly caught species, together with also blue sharks and other big predators like blacktips and tigers. Casao, the second most common species caught by fishermen, is actually a term that is used to refer to many small-bodied sharks, including the smooth-hound weasel sharks, but also neonate and juvenile hammerheads, for example. And it was also interesting to see whale shark make an appearance in the list of commonly caught species, given that it's pretty hard for fishermen working on 5 to 6 meter boats with 15 to 20 horsepower engine to bring a whale shark back to shore. What I want to point out also is that hammerheads and whale sharks are the only actually protected species at the moment in Cape Verde of the ones that are being caught. We do not have, unfortunately, information on what the most commonly caught species in Mayo are. Fishermen did not reply to our question there. But most of them did admit to be catching sharks either regularly or sporadically. And in fact, 90% of all fishermen in all islands reported having caught sharks in the past or regularly catching them. And it is therefore not surprising, but somewhat worrying that 70% of fishermen reported having noted declines in shark populations around the islands. And I've actually noticed the same myself when I went out just a month ago with fishermen trying to find hammerheads to tag. And in a traditionally sharky place where every fisherman will tell you you should be able to catch many sharks, we in fact didn't find almost any shark. We got one in 8+ hours of fishing effort. Perhaps even more alarmingly is that 95% of fishermen noted a decline in general marine resources, so not just sharks, but also the fish on which they rely on for subsistence. And this is, we think, likely due to the increase in population and tourism that is driving the growth of small-scale fisheries. In fact, severe depletion of fish stocks have already been observed in some of the southern islands, most populated island like Santiago down at the south. And fishermen from here have in the past and increasingly are moving to other islands to find fish to catch. In fact, this is probably the reason why in Mayo we are noting this big difference in shark assemblage, as fishermen from Praia have been fishing there for longer, and actually their effect is expanding to Boa Vista. We're seeing more and more boats coming to Boa Vista. So although we have found quite a diverse and locally abundant shark assemblage in all our islands, we're also noting that small-scale fisheries are having an impact on the shark assemblage of Cabo Verde, and increasingly are going to be impacting them unless the growth is regulated. And at the same time, the same can be said for pelagic species. So we hope that with this data, we can help the government create the first national plan for sharks management in Cabo Verde, but also allow us to monitor here in the future, follow trends. And in fact, our next big round of BRAV dropping is coming up soon. We're going to go back to all the islands. Seems like this time I'll have to do some polling myself, unless someone wants to help. Thank you very much. And yeah, thank you for listening.
Thank you, Francisco, for your interesting second speech. We have time for questions.
Okay.
Hi, thank you for the presentation. Maybe I missed this, but have you deployed any pelagic BRAVs or only benthic BRAVs?
It was actually only benthic BRAVs. We have not deployed pelagic BRAVs, though we know people that have. The Monaco exploration came to Cabo Verde in 2017, and people there were dropping pelagic BRAVs, so we're waiting to see what they found and compare it with what we have.
Thank you.
Okay. Thank you, Francesco. Thank you very
Thanks.
much. So we have to postpone the two last talks after lunch, and the keynote speaker Sarah Fowler. But please be here at 2:15 seated. Okay. Now Emilio have to make some announcement.
Just two small announcements. I confirm that this evening, after the end of the meeting, we will have the shuttle that, for who needs, will be able to bring you back to your hotels, like in the morning. And another thing is that we will have also at 5 p.m. a shuttle to della Airport. So if someone needs to go this afternoon, we have 2 or 3 seats more. Good lunch.
Don't forget 2:15 seated, please. Thank you.